Good morning, everyone. Can you all hear me? Can you hear me, Pute? Right. Today I told you I'll be discussing practicals. I gave the tube to some people, but I couldn't give it to everyone. Hashina, if you are listening, just send me a WhatsApp message. I'll forward the document to you. And Manutra I gave, and Dilki also I gave. Ne? Right. Okay, we'll start with the lesson. Right. Let's firstly go to the food test given there. So we'll just go through the given tests. I want everyone to write the name of the test under each category given here. Let's fill the table together. Quickly write the name of the test. You know it now. Wait. Hi. What is the name of the test for starch? Iodine test. Then for reducing sugars, test 1 is enough, right? We are learning only one. That is Benedict's test. Non-reducing sugars. There's no name for that test, but there's a procedure. So we'll be empty, keep that column empty, and we will be writing the procedure later. And then we are going to the proteins. Proteins by unit test. And then fats and oils, Sudan 3 tests. Hi. They will firstly write the test procedure for starch test. So you have to write as it is in the practical guide. So you can open your practical guide and go to the last pages that they have given the procedures there. Yeah, you have to add, I'm talking about test for starch, add 2 cubic centimeters of 1% starch solution in a test tube. Then add few drops of iodine solution. Right, then this is test for starch. Then we will be writing test for reducing sugars, which is the Reddick test. So you have to add 2 cubic centimeters of solution of a reducing sugar. Add the equal volume of Benedict solution. And shake and boil. Fine, write down those two. Then I'll write the test for non-reducing sugars over here. I'll erase the first part, right? I hope everyone is done with the first part. Are you all done? So you are adding dilute HCl here, and then we are boiling. So then only you can hydrolyze the bonds, glycosidic bonds in the sucrose. So those monomer units are producing sugar, so that they will answer to the Benedict test, right? Yan. Cute ka Tharanga nga. Like. So I'm now at this column, okay? Teaspoon on reducing sugars. Procedure I have written it here. Then I'll add the procedure for lipids and proteins. Yeah, firstly write proteins. Next one is test for lipids. How did they finish writing? Hi. I'm going to the next part. It's asking about the color change now. Color change for positive test. So we'll take one by one according to the order we wrote. Um, the first one I would take the starch test, iodine test. Asking the color change there. What's the color change? What's the initial color of iodine? Brown color or reddish brown to blue black coloration, right? So write down the color change. They are asking the color change, not the final color, right? You can't see. No, wait. I'll erase this and write. Can I just? Hmm. Next one, test for reducing sugars. What is the initial color of periodic solution? It's blue in color. Blue color is turned into brick red precipitate. Test for non-reducing sugars also same. Correct? Then test for proteins. Biuret reagent also having copper 2 plus ion. So initial color is blue. Blue is turned into purple. Do you remember these colors? Blue is turned into purple. Next, fats and oils Sudan 3 will give red fat globules. There's no color change, such a thing. Uh, red fat globules. Asking other explanations also under each category. Iodine test, there's nothing much to explain. Regarding reducing sugars and non-reducing sugars, after being hydrolyzed, reducing non-reducing sugars also doing the same. Copper 2 plus ions in the solution, which is giving blue color, which will be turned into Cu2O, brick red precipitate, right? So this 2 plus is turned into 1 plus. So that is a reduction here. This is reducing. This sugar is reducing this compound into this, so that these sugars are called as reducing sugars. Okay. I write down there in that column. Other explanations are not added to the other categories. Now everyone quickly come to the MCQs. There are few MCQs given there from that test. Quickly do that answers there. Hi, this part okay, no? Quickly do MCQs. Hi, are you done with those? Quickly finish those few MCQs here and there. I started discussing MCQ papers now, so I couldn't find a time to discuss and finish it. 2024 paper, second part, I can discuss it tonight. So you can join online for the second part of that means 25th to 50th question, 26th to 25th. I discussed last day. 26th to 50th question tonight. I'll discuss from 9 p.m. to 12 p.m. online, right? I'll put the link into your group also. 26 to 50, right? Okay. You can join. All done? Let's discuss. Mm, right. So first question is asking based on yeah, no, sorry. Test tubes were prepared as given below. Glucose solution, first one. Second one, glucose plus diluted HCl. Third one, sucrose plus amylase. After 1 hour, Benedict solution was added to all three test tubes and heated gently with water bath. A precipitate was observed in which one? What is the answer? Glucose is giving brick-red precipitate true. Then glucose plus dilute HCl also giving, no? So sucrose plus dilute HCl, you know, it's being hydrolyzed. That is not mentioning about neutralizing, but still it is giving an answer. So that is also correct. Next one, sucrose plus amylase, what will happen? Amylase is breaking down starch, neither. Make it correct. I think there's something wrong in starch. It should be starch plus amylase should be. So the answer should be 1, 2, and 3 because starch amylase will give you maltose. Maltose will be answering to this one. Please do the correction in the question. This is an exam question. I have typed it wrongly. That is starch solution plus amylase. That is also giving. So answer is 3, 1, 2, and 3. Which of the following statements is correct? Sucrose gives a brick red precipitate when heated with Failings solution. Failings is similar to Benedict's. You are not learning about failings, but make it as Benedict solution. Sucrose doesn't give, so that it is wrong. Albumin gives a violet color when heated with alkaline copper sulfate. That is true. That is what happening in the biuret test. So that is true. Lignin stains yellow with safrani. We are not learning that. Cut that sentence. Coconut oil is stained with Sudan 3. Yeah. Stain yellow is wrong, but fourth statement is wrong because we have red fat globules. So that is wrong. Glycogen gives yellow precipitate with Milan's reagent. We are not learning Milan's reagent also. Cut that statement. Answer 2 is correct. Albumin gives a violet color, purple violet same, when heated with alkaline copper sulfate. Describe a simple test to confirm the presence of reducing sugars in germinating mung seeds. First of all, you have to say that you are grinding it and you are making a solution from the given sample, right? Without that, you can't do. And then what I discussed in the procedure, you have to add, right? So Benedict test, the procedure steps should be there, right? Next one, question 4 and 5 are based on the following tests and observations. Based on the above observations, it can be concluded that solution X contains, come to X, brick red precipitate. It means it is having reducing sugar. Dark blue color, it means it has starch also. So no color change with biuret, so no proteins. So that you can say that uh, glucose and starch, no? Am I correct? Yeah, answer 1. Based on the above observation, it can be concluded that solution Y contains what? Solution Y. No color change, dark blue, that means starch is there, purple color, protein. Starch and protein only, answer number 1. Am I correct? Right, did you do the next question also? 2025 structure is, sorry, 2005 structure essay. Should I give you a little time to try? Try it quickly. Did you write the answer? Level. So what's asking that question? Hmm. You are provided with three solutions, A, B, and C. One of them contain amylase enzyme and the other two contain 1%, 0.1% and 0.5% starch solution. You are given following materials. White tile, measuring cylinder, stopwatch, water, dilute iodine, test tubes on rack, and glass rods and glass tubes. Let's see. So one is having amylase, the other one is having 0.1% starch, and the other one is 0.5% starch. Now, so you have one white tile, iodine and all. Stopwatch also given, dilute iodine, amylase solution. Yeah, so you can take a little sample of this three of these solutions. You can put one drop each. You can take iodine solution and you can put a drop on that. Neither, isn't it? So which samples would give blue black color with iodine solution? Which samples will give 2 and 3? 2 and 3 starched? Neither. 2 and 3 will give blue black color. So you can obviously identify which solution contains amylase. That part okay? තේරුණාද? හරි. ඒ ටික ලියන්න පිටුර ටික කියලාද? Next week also we will be discussing practicals lamai, right? So I don't think we will have more time to discuss the theory things according to the schedules. On 10th, not 10th, on 11th, I will not be here in the class, but we will be writing a full test there, right? Can you all be ready to 5-hour test to see your performance? හරිද 5 hour test එකක් ලියන්න හැමෝම ලෑස්ති වෙන්න මම ටෙස්ට් එක arrange කරන්නම් ක්ලාස් එකේ මම on time ඉන්න එකක් නැහැ ඔයා ඇවිත් ලියන්න අනිවාර්යෙන් ගෙදර ඉඳන් ලියන්න එපා right okay ඊළඟට කවුද දිල්කී අරවින්දි and all that හෂීනා ගෙදර ඉඳන් ඔයගලන්ට මොන වැඩේ තිබ්බත් 5 hour test එක ඉවර කරලා තමයි අනෙක් classes වලට යන්නේ ඔයගොල්ලෝ දැන් වෙනද කරන වගේ බාගෙට ලියලා පස්සේ වන්නම් අරන් මේව බෑ කලින් ප්‍රශ්නයක් තියෙනවා නම් මට කියන්න මේක ලියන්න මෙපා දැන් අපි දන්නේ නැහැ අපේ performance එක කොහොමද කියලා ඇත්තටම 5-hour test එකක් ලියමු on 11th right so be ready on that I am not sure whether I can have classes after that ඒක බලන්න ඕනේ ඒගොල්ලන් කොච්චර permission දෙනවද කියලා usually exam එකට 2 weeks කලින් අපි නතර කරනවානේ අපි බලමු ඒක මං හිතන් තව එක week එකක් ගන්න පුළුවන් වෙයි let's see about that so that we can discuss the performance as well so please be ready regarding that so you'll be having a complete test there හරි මේක හරියට ලියුවද හරි ඊට පස්සේ you have these two නේ එතන මොකද ඔයා කරන්නේ that you have to you know that the amylase sample now that you will be adding some amount of amylase solution into both the starch, right? So same amount like 2 cubic centimeter making the kai, arakin the kai, hemagan. Arakini will add karala that you will be keeping on a white porcelain tile. These iodine drops are kept. And then another one also. As soon as you mix these two solutions, you will be adding a drop of mixture on top of this iodine solution. You know that starch amylase test, that one. ඒ වගේ තමයි ඔය කරගෙන. මේකේ තිය තිය 2 minutes interval වලින් you will be keeping the solution drop on top of the iodine drop. එතකොට මොකද වෙන්නේ? Initially starch is reacting with amylase, but still more starch is there. Blue black color is there for some time. So, but at some stage, the color will be disappeared, and this will be giving out the true color of iodine, which is the brown color. Brown color එක එනවා. එතන මේකෙත් කොහොම කරගෙන යනවා. ඔය දෙන්න ඔයගොල්ලන්ට හිතෙන්න ඕනේ දැන් which one would stop early? Which is having high concentration? This one or this one? This one is having high concentration. So one which is having high concentration will take longer time to finish the reaction, isn't it? Neither. එතකොට ඔයගොල්ලන්ට identify කරන්න පුළුවන් කෝකෙද 0.5% තිබ්බේ කෝකෙද 0.1% තිබ්බේ. තේරෙනවද? හරි. හදලා ලියන්නකෝ මම දෙන්න එකකින් එකට හිතලා ලියන්න. Right? So now you have 2 weeks remaining now, so please study well for the test and get final idea about your mark, okay? All done? Shall we go to the next section? Right. Next section is about microscopes. I think you know how to label the parts of the microscope, isn't it? Right. Right. Skip that first question. What are the instructions you can give for safe handling of light microscope? And please go to the next question and everyone please try. Then I'll write my answer. Then we'll discuss together. And quickly do the questions, MCQs also from microscopes. After writing the answer there for this one, what are the steps that you would follow in order to observe onion epidermal peel under the mid power of the light microscope? Quickly write the answer for that one, everyone. Alright, are you done with it? So question is asking you to write the steps now. So what is being done first? Number one, you have to peel. Peel and take an onion, epidermal peel, and place it on a watch glass of water. Alright. Number two, take a clean glass slide, place a drop of water on the glass slide using a paint brush. Alright. And number three, select a මෙතන එකක් නෙවෙයි, let's take a few from that you are taking one here. Take a thin onion epidermal peels and place on the water drop. Then number four, cover with a cover slip. Number five, remove excess water on the slide using a blotting paper. Or paper serviette also you may say. And then you are asked to use the microscope also. No, that step also, that steps also you have to mention. Number six, keep the slide on the microscope stage. Then number seven, secure it with stage clips. Then you have to adjust the mirror. You can't skip these steps, right? Adjust the mirror. To get enough light to observe the specimen. I'm erasing the first part. Are you writing this one? No. Then number 9, observe using low power and rough focusing knob is used to adjust Number 10. Turn to mid power once a clear field is observed, obtained from low power. 11. Adjust using fine focus now. Alright, write down and keep. Alright, done. Did you do MCQs also? Alright, let's discuss the MCQs. 1 minute, please. Alright. The first MCQ. We have some remaining MCQs. Oh, wait. In the previous one also, I have skipped and went. Right, let's come to that one also. Which of the following carbohydrates give or give a positive test with failings test? Failing is similar to Benedict. Cut that and write Benedict's there. Uh, Glucose is giving an answer. Fructose is giving an answer. Maltose is giving an answer. Lactose is giving an answer. So that answer is 5. So only sucrose is not giving an answer to failings test or Benedict test. Which of the following carbohydrates gives a positive reaction with Benedict test? Lactose, glucose, maltose, ribose. Again 4, 5. Answer is 5. Right? Which of the following chemical test can be used to show presence of glucose in a solution? Benedict's test. 2. Then there's another structured essay here. Quickly we'll go through that one. Name a laboratory test used to identify the following. Main type of organic compound found in egg white, biuret test. That is protein. No? Answers. Biored test. Next one. The main type of organic compound found in coconut milk. Sudan 3 test. Next one. Main storage substance of chlorophyte, that is starch, therefore the test is iodine test. Reducing sugars, Benedict's test, right? State the common characteristics of all monosaccharides and some disaccharides. These are reducing sugars. That's the expected answer here. Next one, you can do it now. Let's go to the MCQs from this one, the microscopes. Which one of the following cannot be seen under light microscope? Plasmids, answer 3. What are plasmids? Addition to the main chromosome in prokaryotes, there are some small DNA which are circular that are called as plasmids, having few genes there. Next one. A student was provided with an epidermal peel of onion mounted on a glass slide and a light microscope. State in correct sequence which steps should be followed to observe the shape of an onion epidermal pieces under light microscope. You have to write the answer what we discussed now. Which of the following statements regarding microscopes is correct? In light microscope, visible light is passed through the objective lens and then through the specimen. Wrong, ne? Firstly through the specimen and then through the object. Ne? Or projection of light beam through a vacuum is principle of electron microscope. No light beam, it's electrons. Ne? That is also wrong there. Scanning electron microscope is used to study internal structure of the cell. No, surface structures are studied using scanning electron microscope. Transmission electron microscope is used to detail studies of living specimen. No, it's dead specimens. Number 5 is the correct answer there. Magnification and resolution power are important properties of all microscopes. Okay, children? Right, next one. Steps involved in observing an onion peel mounted on a glass slide and placed on stage of a compound light microscope are as follows. So what do you do first? Adjust the mirror. Mirror should be adjusted to get enough light. That is the first thing to do. So A is there. And using the coarse focusing knob, you can find out the answer from this one as well. Then using the fine focus last. So A, C, B is the answer. Answer 2. Okay, everyone? Right. So I have given few pictures down there of electron microscopes, the scanning and transmission electron microscope. I want everyone to do the questions coming up next. That there are some pictures of chloroplast, ER, Golgi, mitochondria, nucleus, and bacterial cells. And yeah, few pictures are there. You have to figure out whether these pictures are from scanning electron microscope or light microscope or transmission electron microscope. And everyone, page number 9 and 10, there are some comparisons. Quickly do and finish off that comparisons. Will be given little time. Quickly do. We have to go to the other part as well. All done? Did you write the differences also? Anita, ivarai. Aravindi, Devaki, done? Right, let's go to that one. Ashina, are you there in the class? Right. Here, all these pictures are taken from transmission electron microscope. Can you understand? Why is that? Why am I telling that? All these pictures are two-dimensional pictures which are taken from an electron microscope. Light microscope, you can't see much details like that, isn't it? Neither. Let's try the differences between light microscope and electron microscope. Can you all go to the table given there in the book? There's a table with differences between light microscope and electron microscope. Can you all come to that page? Do you have the book? Book grade 12 theory book. Grade 12 theory book. Under microscope, they have given a table there. Come to the table. Let me tell you the page. Page number 24. Did you find it out? Yeah. So there the differences are given like this. Light microscope, glass lenses are used to focus the light rays. Electron microscope, powerful magnets are used to focus the beam of electrons. Light microscope, image is directly detected by naked eye. Electron microscope, image is not directly detected by naked eye. Micrographs are used. And living and non-living objects can be observed in light microscope. Only non-living objects are observed in electron microscope. Next one, actual color of the object can be observed in light microscope. Actual color cannot be observed in electron microscope. Next one, dyes are used to stain the object, light microscope. Heavy metals are used to stain the object in electron microscope. Right, so see the differences. Then the differences between SCM and TEM. SCM, surface details of the specimen is observed. Here, internal structures is observed. And then here, SCM is three-dimensional view of three-dimensional image is created. Here, 2D image is created. In both the cases, heavy metals are used to stain. All right. So those are the two major differences here. Animal cell and plant cell differences. What do we write? Major differences only we will write. Hmm? What are the major differences? Here, central vacuoles are absent. Here, large central vacuole is present. Then number one is that. Number two, um, here glyoxysomes absent, glyoxysomes present. And you can say number three, centrioles are present. Centrioles are absent. Right? Number four. You can say chloroplasts are absent. Chloroplast present. Haripathe? All right. Next, anything else to write, Puthe? Anything I forgot? That's all, eh? Hi. Let's come to this next section about mitosis and meiosis. Can you all come to that page? I want you to do the small questions quickly from one. Second one, if you don't know, leave it and go. Other questions, please quickly do. There are small questions present. All right, do that quickly, do right, so that we can go to some more practicals. Finish with it? All done? Ah. Adipati, can we discuss? Right. Aravindi done? Hashina? Feel clear? Right. So here, the first question is asking what is commonly used tissue to observe stages of mitosis? Onion wrote it, right? Then next question, you can identify few stages. If one cell contains two nuclei like this, that is telophase. If a cell contains chromosomes like this, that is anaphase. If a cell contains chromosomes in the middle like this, we call it as metaphase. Chromosome with prominent, it's a cell with prominent nucleus, with nuclear envelope and nucleolus. We call it as interphase. Correct? If a nucleus and nuclear envelope is available, chromosomes visible as chromosomes like this along with the nucleolus, prophase. Next one, meiosis results in genetic variation through recombination of alleles, independent assortment, and crossing over. Meiosis consists of two main stages, stage 1 and stage 2. Put a cross in the box next to the phase of meiosis when independent assortment takes place. Answer B. Metaphase, sorry, not B. Metaphase 1, answer A. Independent assortment occurs in the metaphase 1. Do you know what is meant by independent assortment? Hmm? What is meant by independent assortment? In the metaphase, the random arrangement of chromosomes, we call it as independent assortment. Let's say, I'll take another color and come. Another chromosome is there. Homologous pairs are randomly arranged on metaphase plate. We call it as independent assortment. This is one way of arrangement. There are so many other possible methods of arranging these chromosomes, orienting these chromosomes to different poles. So many the number of chromosomes, many number of possible combinations are there. This is called independent assortment. This is also talked in Mendel's which law? Hmm? In Mendel's which law we are talking about this one? First or second? Second law. Mendel's second law is talking about independent assortment. They are telling alleles are segregating independently, assorting independently. Describe how independent assortment results genetic variation. You can say that independent assortment results in new genetic combinations which will lead to new genetic variations, which let's say it may or may not, right? New genetic variations and it is not a must, right? It might lead to evolution. All right. All right. Next question asking, put a cross in the box next to the phase of meiosis when crossing over takes place. Prophase 1, answer C. Asking what is the stage given in the diagram, that is metaphase, answer B. Put a cross in the box, correctly identify the stage in which DNA is replicated. Interphase, answer B. In the interphase, S phase only the DNA replication would occur. Right. Did you recognize the next phases? Identify karada? Nane, karan. Taggala karan. Kadavutela. Right. So here, what is the given first stage? This is anaphase 1. First diagram on the left side, anaphase 1. Second diagram towards right to that diagram is anaphase 2. Then third diagram is prophase 1. Fourth diagram is metaphase 2. That is the bottom one from the second line, left side. And the second one towards right, that is metaphase 2. And the third one, it's telophase. No, the telophase 1 it should be, because complete chromosome is there. It is not the correct way to draw this one. You have the network of chromatins here, right? All right. Next, go to practical number 5. I'll give you a little time to answer the short questions given there. Quickly try, then I'll explain the experiment in 5 minutes. Quickly do answer to the small questions given there. Okay, shall we quickly discuss? Right, so here, first I'll draw what's happening in the experiment and explain a little. Uh, we're taking amylase sample and starch sample. Both will be kept in water bath having the relevant temperature. Then you need to mix these two solutions according to your book. Let me see what is next to what. Correct way is not what is given in your book. It's given correctly in Sinhala book. You're mixing amylase to starch, okay? Understand the correct method. So you will be keeping it also in the water bath, and you're taking a white porcelain tile which is containing iodine drops. And as soon as you mix the mixture, you'll be adding 1 drop of iodine on top of this one. You will observe blue-black color. So it will disappear after each 2 minutes time interval. You'll be keeping this drop, and after some time, the color will be disappeared. That the true color of iodine will eventually form because all the starch is hydrolyzed to maltose, right? Then another, yeah. Here you will be taking amylase and starch. This is amylase is the enzyme here. Starch is the substrate here. You'll be keeping both of them to reach an equilibrium. That means, let's say if you are using 30 degrees Celsius, that particular water bath, you will be keeping those two solutions before it start the practical. And then after that, you'll be mixing these two solutions. This is the correct way of mixing. You are adding amylase to starch solution, right? And as soon as you mix the mixture, you will be placing a drop on top of iodine drop, and the color change will be observed. So initially, the amylase is not converting starch at the beginning. So if the starch is already there, starch is giving a color change with iodine, that is blue-black color, right? Right? This blue-black color will be there at the beginning as soon as you mix the mixtures. So as soon as you mix the mixtures, you will be starting the stopwatch too, so that you will be testing drop from the relevant solution in each 2 minutes time interval. හැම 2 minutes වලම බලනවා. බලද්දි blue black color එක තියෙනවනේ ටිකක් දිගට. blue black color එක තිබිලා after some time the blue black color will disappear. Disappear වෙන්නේ මොන වෙලාවේද? All the starch has to be converted into maltose by amylase. Right? This is the enzyme amylase is converting this one into this. So if maltose is present, you can understand there's no more blue-black coloration. So that after some time, after particular time period, you will get this is initially blue-black. Here it is brown color of the iodine will appear. So you'll be recording that time in a period that is taken for the color change to occur. Then another, draw this one in the cage given there. Done? Alright, let's go to the first question. What is the enzyme that we use in the experiment? What is the enzyme? Amylase. Write that there. Number 2, what is the source of the above enzyme? Saliva is the source of the above enzyme. What is the substrate for the above enzyme in the reaction? Starch is the substrate. And the next one, how do we bring both enzyme solution and substrate solution to the same temperature prior to mixing? Keeping in a water bath containing the relevant temperature. If you ask this question other way around, you should have to be able to answer now. You are keeping in water bath. It's in order to achieve both of them into the same temperature. Right. Next one. Why it is necessary to bring both into same temperature? What is the reason why we are taking enzyme and substrate into same temperature? Is there a reason? Otherwise, what will happen if you are checking the effect of temperature? If the both two solutions are in two different temperatures, that will affect the rate of reaction, right? Isn't it? Right. So the answer is if enzyme and substrate will be on two different temperatures, it will affect the rate of reaction. Right. Then next question. Which solution should be added to which? Amylase to starch or starch to amylase? What did I tell you before? Hmm? Amylase to starch, yes. Right, good. Next one. Uh, why we should start stopwatch as soon as we mix the solution? Because as soon as we mix, the reaction is starting, right? As soon as we mix, reaction is starting. That's the reason why we use this one here. Next one. What is the solution taken to test this reaction mixture? Iodine solution, or as you can write I2 in Ki solution. Next one. Why a white porcelain tile is used? To observe the color change clearly. To observe the color change clearly. Next one. What is the color that you observed in a drop of mixture taken as soon as enzyme and substrate mixed? Blue-black color. What is the color that you observe once the reaction is over? Brown color, iodine color, right? 12th question I need you to answer at home by looking at the experimental setup, experimental data in the given book. All right, 13. Quickly draw the graph that you expect here. That if you are changing, according to the previous question only you had to write, the effect of temperature on the rate of enzymatic reaction. Culture, no, probably. Right, give me a few minutes to start, yeah. Yeah. All right, it's okay, it's okay. Did you bring it today? Okay, good. Yours also, you can keep it here. Right. Others, what is the graph that you are getting here? This side it is temperature. This is good for you guys that you also have not done the graph properly. Rate of reaction. Graph right. So here at the beginning, the graph goes like this. That is an acceleration, but there's a deceleration too at this place, right? Ogalonge graph වැරදි බලා ගන්න හොඳේ. සමහරුන්ගේ වැරදි. So this is the optimum temperature. You have to mark the optimum temperature over here or here. Here, after that, there's a rapid decline. It should look like this. Some have done symmetric graphs also when I'm marking the papers. That is wrong, right? This is an asymmetric graph. May patta lokui, right? May patta podi.